Do you have a tendency to be slightly OCD when it comes to cleaning? If so, I highly recommend you to become a molecular biologist. Today is day 2, and we have spent pretty much our entire day cleaning. And when I mean cleaning, I mean that there's literally nothing organic left, not even a strand of DNA on any of the gear we're going to use for our sampling. And that's exactly the point. Not a single strand of DNA left. Because you see, what we're going to be doing on this ship is molecular biology. And what we're interested in getting from our samples is DNA. We're going to be taking water samples that potentially have DNA floating around in them, and if we have any kind of other DNA hanging out on the gear that we're using, on the lab consumables, on the bench, that's contamination. And the thing is, we're on a research vessel where people are going to sample zooplankton. They're gonna trawl for fish. There's all sorts of sampling of marine organisms going on, and the DNA of these marine organisms is just everywhere. It's in the ambient air. We breathe it. I breathe marine mammals. So it's really a problem when you actually take water samples and you're just wondering, I wonder what kind of DNA I have in this sample, and then you've got all this DNA from the outside that just kind of falls into it, and you think, oh, look at that, I had, in the ocean there, what did we have? We had sharks, we had this and this species of fish, we had zooplankton. Oh, wait a minute, we have Homo sapiens chicken. So we can't. That's not possible. That's contamination. And we need to be able to account for that. And that requires cleaning, more cleaning. And because today was the first day where we were setting up the lab space, we cleaned everything. We bleached everything, because bleach degrades DNA, so it's actually really good. The problem is that it's a little bit too good. So then it might actually degrade the DNA in our own sample, which we also want to try and avoid. So we need to find the right balance between super clean, but not to the point where it's also going to clean our own sample. So that was today, really. That's what we did. It was a very, 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 very long doing the dishes session, basically. And if you want to do this kind of work, you do have to enjoy doing the dishes, because unfortunately, it doesn't happen just when you set up the lab. It's gonna have to happen after every single sample you take. And when we have about 1 to 2 hours of processing of sample, we have another 2 to 3 hours of cleaning. So please join me in the Mega OCD cleaning team, aka the molecular biologists.